And this is David Roos. As Omar is getting ready to get us launched, I will just mention to those who are here already that I suspect that there are a lot of slightly different agendas that people have on the call, a variety of questions that people may have about ToxoDB or about annotation or about reference genome assemblies and about new and emerging data sets. We're happy to have all of those conversations, even individually or offline. But today we're going to focus on we'd like to focus on our efforts to try to capture expert knowledge from the community to improve the reference gene annotation as opposed to genome organization for Toxoplasma. That's not to say that we're not interested in having all of these other conversations. This will not be the only opportunity or venue to have this discussion. So feel free to get in touch with us at any time by clicking on the Contact Us link at the top of every page on ToxoDB or ViewPathDB. Actually, I guess hidden underneath the slide that I'm sharing right now, maybe you can make out a little bit of the Contact Us up here.
Okay, I think we should go ahead and get started. This webinar is being recorded, so obviously others who are not able to join today will be able to watch it if they want, and of course provide their input by email, which would be great, obviously, too. Okay, so we have a few things we want to cover today. David is showing a slide with today's agenda. I think everybody's been looking at it. So we'll discuss a little bit about what are we really trying to accomplish here with the community annotation and updating and improving the reference annotation. As hopefully everybody can appreciate, this is not only a problem or an issue for the Toxoplasma genome, but all genomes face issues with their annotation, both the structural annotation of genes and the functional annotation, the description of those genes. Both of these are issues and considerations, and we would like to discuss sort of our goals. Another topic is to discuss the reference genome assembly and thinking about how are we going to work on updating this. As most of you obviously know, the genome, the assembly, at least of the reference genome ME49, does not have the right chromosome numbers based on new data that came out from multiple groups, many people represented here on the call. And we want to make sure we are using the correct assembly, but there are obviously caveats to all of this. We'd like to discuss those. We will also highlight the annotation updates that have been done through ViewPathDB, through ToxoDB, using the community annotation, the Apollo community annotation system and user comments. And so we'll highlight some examples, and then we'll talk about a timeline of next steps. We have an item on the agenda for questions and concerns and open discussion, but I think we all know each other very well here. The community knows each other. So feel free to unmute yourself at any time and butt in with a question, or use the reactions panel in Zoom to raise your hand, and we will call on you to talk if you prefer doing that. But at any moment, if you have a question or confusion about something or need additional information, don't feel like you have to wait all the way till the end to ask your question. David, maybe you can move to the next slide. Okay, so in terms of the assembly status, I'll just summarize this, and I'm sure David will have additional things to say. So before, actually, one thing I forgot to mention is that in addition to me, I'm just going to be talking very little, and I'll take some notes in the background. But David and Uli Böhme, who's on as well, will be covering most of the webinar and really delving into some of the issues and concerns and genome updates. I think with the assembly status, we're aware, again, as I mentioned, that the current assembly has issues, but there are new assemblies out there. Now, the question is what do we do? And the three options, and maybe there are more, and we're open to suggestions, is one thing is we don't do anything with the assembly. We just keep the current assembly, And again, just as a reminder, we load all the genomes, right? So any genome that has annotation, even if it doesn't have annotation, we can load it, but obviously much more useful if it has annotation in some cases. But we load all of these genomes, and so we can load multiple versions of a genome. This comes with its own problems, of course. But one thing we can do is we don't make any changes to the current reference genome in terms of the assembly. The second option is that we select one of these new assemblies with the correct chromosome numbers, and we make that the reference, the new reference assembly. But then we have to make some decisions about multiple things. You know, how do we annotate it? Do we take the new annotation from that assembly and make that the reference annotation, and then we update that? Do we transfer the annotation we have from the current reference annotation to the new assembly? What do we do about gene IDs? How do we track them? And many other concerns. We can potentially, as another option, harmonize the existing assembly into something that's a consensus, but a consensus assembly from multiple assemblies and annotations. I think this refers to something that often is called maybe a pangenome, but this comes with lots of caveats and problems, and lots of people have worked on trying to do this, but it's you know, there aren't any. I don't think there are any great examples of this being successful. But you know, this is an option we can certainly consider. We can support multiple reference genomes. So the reference genome, again, in ViewPathDB, it's really what we use internally as the genome that we will map all functional data to. So all the RNA sequence data, the proteomics data, we kind of map everything to one reference genome, so you can go and ask questions about genes in the genome and look at all the data, even if the data was generated in RH or VEG or any other strain. It can be mapped to the MB49 strain, and so everything is harmonized into one assembly. But we can actually map the same data to multiple assemblies and multiple genomes, and so that's another option. And so we can support multiple reference genomes, and of course, you may have additional suggestions. So David, I'm going to stop here with what I wanted to say and turn it over to you for further discussions. And again, as a reminder, if you have questions, feel free to raise your hand, unmute yourself, or you can type them in the chat as well.
Thanks, Omar, and Uli, who is a curator and annotator on the project here. Just a note to you, I'm not sure if I wound up reordering the slides a little bit. You know, when we turn to the Apollo demo, I'll certainly turn it over to you to share. But the next slide that was included here was a summary of existing information, you know, already available within ToxoDB, ToxoDB component of ViewPathDB. I'm not sure if you want to come back to this later on, but I'll simply point out that currently a large fraction of the Toxoplasma genome does have annotation changes that differ from the reference that's in GenBank and differ from the original annotation that was available on ToxoDB. And Uli will provide some indication of where those changes are and how those changes can be made. And there are also hundreds of genes with updated functional annotation, as well as thousands of user comments that are attached to individual gene pages on ToxoDB. So I don't know, Uli, if you want to say anything more about this summary at the moment, or shall I just highlight what the problem is through a couple of JBrowse views?
I can say later a bit more when I do the Apollo demo.
Okay, so that's fine. So you may have the slide, or I can pull it up later on. But this is really just to provide in the community with an indication that we as a community have quite a lot of information that is currently available on ToxoDB and is highlighted in ways that allow you to look at those means. As Omar mentioned, there are obviously ways in which this will change with an updated reference genome assembly, taking advantage of the long read sequences from work in Maria Francia's lab and from John Boyle's lab and perhaps others as well. But their nice work has indicated that this will not affect most of the individual genes in the genomes, although it affects some genes that many of us care about. And so our goal today is primarily to focus on individual changes for genes. So let me highlight what some of the examples are in just 5 minutes before turning it over to Uli to give you a hands-on demonstration. I'll do this just in summary form and in PowerPoint slides, but it all illustrates functionality that I think most of you will be familiar with accessing. We won't have time to go into the details of how I've configured these browser views, but there are a variety of tutorials that are available under the help window on ToxoDB, and of course, we're also happy to take any questions at any time by clicking on the Contact Us link. So I'll start by illustrating the problem, looking at a gene page for the HXGPRT gene in the reference ME49 genome. And I highlight this gene, which many of us are familiar with from selectable markers, a gene that was actually cloned in my lab a generation ago, as a selectable marker. But the reference annotation for this we know is wrong. This was the first gene identified as being alternatively spliced in Toxoplasma. And in the original publication of that, we had noted two polymorphisms, the presence of an exon skip polymorphism that includes or excludes an in-frame coding sequence that's functionally important because it encodes an acylation domain that leads to membrane association of this enzyme, but also an intron read-through polymorphism. And so technically, these could be included or excluded, giving rise to 4 possible transcripts. But the reference annotation here is now a generation behind what we knew from this publication in wherever, JBC. If we scroll down the page to take a look at the vast wealth of functional genomic information that is now available on subcellular localization and essentiality and so on, we can see a summary of transcriptomic views here indicating that that this gene is most abundantly expressed in the cat stages that we had no information on a generation ago. These from studies in Adrian Hell's group, and that'll come to be useful if we zoom in to take a look at or move to take a look at the in the genome browser. I have highlighted the official annotation here in the yellow track. This is not how it will show up, and the vast wealth of information that is available. If I click on selecting tracks, you can see that there are somewhere here indicated, yes, up at the top, many, let's see this out of the way so I can, that there are 761 tracks of information, which you might choose to pull up. And I have pulled up a few selected ones, mostly of historical interest, highlighting the organization of the chromosome, which in this case is well assembled, no gaps that are likely to interfere with it. I'm doing nothing unusual in GC content or low complexity regions or tandem repeats. The original annotation was exactly that was shown in that JBC paper. A variety of automated gene models suggested possible alternative annotations. The existing annotation has been revised to highlight non-coding sequences, and this is all consistent with what we saw from the old EST data sets, which show both read-through in this in the first UTR intron, and also the presence or absence of this in-frame exon skip polymorphism. This is also supported now by long read cap selected data, which highlights the most abundant transcript here as being something in the middle, highlighting that in fact something we didn't fully appreciate previously, that prediction of intron read-through in the first intron was inaccurate. And in fact, there seem to be 2 predominant transcriptional starts here. And of course, if transcription starts within the first intron, that intron cannot be excised, but that has no impact on protein coding sequence, which begins down here in the second intron. This is also supported by proteomic data from many experiments, collapsed now here into a single track, and most prominently, of course, from RNA-seq data, all of it summarized, but I have pulled up from those various tracks that particular experiment that we saw on the gene page that showed the most abundant transcription in not just in tachyzoites and bradyzoites, but in the gametocyte stages, where we can see even higher expression. This is now shown on a log plot, so this level of expression is probably an order of magnitude higher than what is seen here in bradyzoites. RNA-seq data also allows us to extract information on intron-spanning reads, and this is particularly useful in something that I believe is only represented on ViewPathDB databases. I'm not aware of any other databases that collect this information, although individual research labs have. And it allows us to see that, for example, these 3 annotated introns are indeed supported by tens of thousands of reads, so very strong support for the existing annotation. You will note, however, that introns 2 and 3 are represented by only 10,000 reads as opposed to intron 1, which is represented by 50,000 reads. And that's because there is an even more prominent unannotated intron that drops out this acylation domain and is represented by 40,000 reads with abundance that's consistent with what's known from proteomics data and Western blotting and immunoprecipitation. There's a great deal of other information that we won't have time to go into today, but which can inform your assessment of annotation, including epigenetic data from ChIP-seq and ChIP-ChIP data sets, comparative transcriptomic data, genomic data from SNPs, comparative genomic across species. I've selected individual tracks to show Neospora and Camandia and Sarcocystis as well as Eimeria. But the most informative information is certainly these intron spanning reads, which allow me to recognize that this should be re-annotated certainly to include at least the it's on skip polymorphism. And if we go back to that view, that change can be made, in fact, is available today with an alternative that has been added in a community annotation track that Uli will take over to describe how to do in just a moment. In this particular case, we haven't annotated this alternative start or multiple alternative start sites for this transcript, but perhaps that should be added, nor have we indicated alternative stop sites here, which are present, remember, on a log scale representation at less than 10% of total transcript. But this might be important to you for functional reasons and other, and I'll use this point to illustrate that that many other changes have been made. Someone, quite likely Uli, possibly me or someone else, has corrected the annotation of this gene in which the exon structure is correct, but to more accurately reflect the proper start site, and highlighted that the likely translational start site is downstream of what was annotated here, consistent with the fact that some transcripts can be longer. Moving to the right, we'll see a region where there is no annotation for understandable reasons, because we now know that this transcript is only clearly evident in gametocyte stages, but that annotation has been added. And finally, if we we can also see evidence of other features, including a low abundance but consistently observed opposite strand, probably non-coding RNA, which again one might want to be able to annotate. There are many other more complicated regions moving downstream to this. We can see another region with a gametocyte specific and that shows transcriptional changes, transcriptional initiation sites here and here, coupled with a long non-coding RNA, some of which might be appropriate to annotate, but we can see that at least these 2 transcripts have been annotated. So with that, let me turn it over to you, Uli. You're welcome to share your screen, or if you'd like to just ask me to advance, I'm happy to keep this up, or you can do whatever you like.
I would like to share the screen to give a short Apollo demo of about 10 minutes and to show how you can change gene models in Apollo.
Go right ahead. You should be able to share your screen now. And I'll reiterate that we will certainly leave time for questions at the end. If there are any burning questions now, feel free to raise your hand, speak up, or type it into the chat. And Omar and I will be monitoring the chat while Uli is talking as well, if people have questions they'd like to raise at this point.
Thank you, David. I hope everybody can see my screen. So what I'm going to do now is I'm going to show you a little bit about Apollo. Omar already mentioned that. So Apollo is an extension to JBrowse, whereas in JBrowse, as David showed you, you can look at the gene model. What is missing is you cannot really change a gene model, or you cannot add a new gene model. And that's why Apollo has been developed, and we offer that on our sites. And I'm going to walk you through 2 examples just to show you how intuitive and how easy Apollo is in usage. So the first gene model I'm going to show you is this one here. And when you look here on the gene record page at these unannotated intron junctions, what you can see here that those 2 were not considered in the current gene model. And this is very easy to fix in Apollo. So you can access Apollo from the gene record page, either from here, or you can go to the tools menu and go to the Apollo site. There is additional information here. For Apollo, we have tutorials, webinars, screencasts in case you're interested. Here's the link to the live Apollo site. This is another Apollo instance, which we use for demo purposes and for learning purposes. So if you're new to Apollo, I would urge you to have a look at the sandbox site, and you can play around with Apollo as much as you like. And for this demo, I'm also going to use the sandbox. Once you click on the sandbox, what will come up is a FewPathDB login. And I just would like to mention that you don't need a separate registration or any other login for Apollo. So you just need your FewPathDB, ToxoDB credentials, and you can use Apollo. So really easy. I'm already logged in, and this is the gene model I've just showed you on the gene record page with these 2 unannotated intron junctions. So this might look very familiar because this is really JBrowse. So you can select tracks, similar to as David showed you, from this menu. Apollo has a separate menu here where you can also select tracks. And this is what I'm going to do now. The first tracks that are always important in Apollo are these trackable annotations. I'm interested in the current annotation, and this looks a bit different because Apollo gives you the different frames. So this is why it's a bit colorful. I'm also interested in introns that match the transcript annotation and introns with strong annotation. To know how many reads I have, I always use this drop-down menu to show labels. So this is not important anymore. I'm going to get rid of this. You can use the search menu to look for additional evidence, and I'm interested in this. Okay, so now let's fix this gene model. There's an exon missing, clearly visible here. What you need to do is you need to drag up this gene model into this yellow area, the user-created annotations area. To do so, you just click on the intron until you see a red border, and then you can just drag it up into the user-created annotations area. Or alternatively, with a right click, there's a menu, and you can say create new annotation and gene. It's really easy to add a new exon. You just select the unannotated intron junctions with the shift key. You can select both, and then just drag them up in the yellow area into the gene model until you see a green border. And the rest is also easy. You just need to fill in the exon, and you can do that by either selecting both of them with the shift key. With a right click, there is a menu with different options, and one of the options is merge. One other option is go to select one of the exons and hover at the edge until you see a little arrow, and then drag it up into the other one. So an exon has been created. What I also would like to do now is add UTRs, and you can see here we've got evidence that this can go longer, and this is as easy as adding an exon. Hover at the edge, select an exon, and drag it up until the end. Yeah, a bit too long. And the same here. Select the exon, hover at the edge until you see a little arrow, and then go up here, and you can zoom in very easily. You see this gene model sequence, and then go to the end, and you can explore it further. So this is how easy it is to add an exon and add UTRs. And the last step you need to do if you want to correct the gene model is select the gene model, use a right click, and then open the annotation. And then you come here to a menu that gives you options to add function annotation, go terms, gene products, and dbxrefs. What you can also do is then select the status. So once you are happy with your gene model, you just select finished. And this is where the pipelines then look for this. There's a pipeline that looks for finished annotation. And the next day, this gene model will show up in the community annotation tracks that David showed on one of his screenshots. And I'm going to show you one more gene, similar to JBrowse. You just need sorry, wasn't too fast. This one here. Okay. And I'm not interested in this gene. I'm interested in this one here. And as before, you can see there's evidence and there are unannotated introns, but no gene model. And it's really easy to create a new gene model. What you do is just select the intron junctions. You can drag them up into the user-created annotations area, or with a right click, go to the menu and say create new annotation. This adds it up into the yellow area. And as before, I'm just now filling in the exons. And you just use this and drag it. Same here. Just going from one to the other. And finally, I want to have methionine and a UTR. And the fantastic thing about Apollo is it does a lot of the work for you. So it looks for the longest CDS. So it looks for the methionine, the longest CDS, and adds then also the UTR. So really easy to use. And I'm going to also add a UTR here. Zoom in and out a bit. So, and go up to the end. And so this is how easy it is.
Can I ask a quick question?
Of course.
Okay, so I see that you're modeling this gene from a series of introns that are supported by reads, but often what we're doing is we're starting that with just individual introns, but reconstructed transcripts. Can you start with reconstructed transcripts here and just drag like a whole transcript up into the panel?
Yes, yeah. I mean, you can. But as I showed before, you can just drag up. Does this answer your question? You can drag up.
So Uli, I mean, I think so. The issue is so the draggable tracks that Uli is showing here need to be created within JBrowse, and we can create those tracks quite readily. So if there are reconstructed transcripts that you'd like to provide as a GFF, that's something that can easily be entered into the system to be able to add that. But I don't believe that it is that Apollo currently supports in the adding a reconstructed transcript model from the GFF without a draggable track. Is that correct?
Yes, well, we've got only these, the ones I've showed you as trackable annotation, but this certainly can be done.
I'm just saying, if Brian has a does that answer your question, Brian?
Yeah, I'm just wondering if you're starting off with something like string tie reconstructions,
Yep.
Or maybe like Trinity alignments, something like that. You could use those as starting points.
Yeah. So there's a lot of information, and this is something we certainly. So if there's particular, I mean, as you've seen, there are many hundreds of tracks that could be useful. The intron spanning reads is all aggregated, and we have done advanced work to highlight what the abundance is in various different views of those that are of highest, medium, or lowest confidence, and those that match existing annotation or do not. That's what Uli is using now, but it would certainly be, and I don't believe that Apollo supports the entry of a gene model directly from a GFF3 file of a reconstruction from, say, a Trinity reconstruction. But that's certainly something that we could explore, and it would certainly be an easy matter to provide a draggable track like the draggable tracks that Uli has shown.
Perfect. Thanks.
Thank you. So just to finalize this, what you can do if you create a new gene, use that drop-down menu to get the sequence, and then you can explore it further. Go to Interpro or Blast.
I'm sorry, Uli. Dave Starnes entered a note into the chat indicating that local tracks can be loaded and configured as draggable. So thank you, Dave. I didn't realize that individuals with, I mean, I'm familiar with loading local tracks into the JBrowse browser. I didn't realize that they could be configured as draggable within Apollo. So that's something to maybe follow up on with Brian or anyone else who is interested. Brian, if you want to know more about that, click on the contact us link, send us a look, and someone likely Dave Starnes will get in touch with you.
Okay, just would like to finish this gene model. As before, just open the annotation and select status finished, and you can add additional annotation, function annotation. And this is actually all I wanted to say for now about Apollo. So if you're interested to learn more, please get in touch. Also, if you have tracks, and please get in touch with us if you want to learn more about Apollo. And what I would like to do now is just give you a summary of the changes that were already done with examples that were done. The majority of the changes are structural changes. And of course, the majority of them are UTR modifications. And David already showed the numbers. I'm not going into detail, but we have different kind of CDS modifications. And the majority of them are exon changes. We've got genes that are too long or too short. Genes that look perfect, but they are starting on the wrong feeding frame. We've got merged genes, quite a high number. Also 258 new genes, and the majority of them are protein encoding genes. We've also got genes where the gene is completely on the incorrect strand. And we also have now genes without any evidence. And because of all of the changes, they are now overlapping with new gene annotations. And we've got isoforms, and one of them David already showed you. And I'm going to show you an example for each of those changes. So here is the highest amount of changes, which is just UTR changes. So in this case, an addition of a UTR. And you can see it's nicely supported by transcription start site. Then we've got also nanopore that supports this. So we've got over 2,000 genes that have a UTR modification. Then we've also got, and this might look familiar because this is the gene that I've just showed you and that we've corrected in Apollo. So this gene has these additional exons. And I'm highlighting this gene because this highlights quite nicely that every single exon counts. Because without the exon, there is no interprodomain. If you add the exon, you've got a nice interprodomain, and it's a selenium protein and not a hypothetical protein anymore. Another option of changes are that over 200 had a shortened first exon, and David also showed you one of those. And you can see here, nicely supported by transcription start site, this is certainly was too long in the current annotation. Then we've also got here extensions. So around 70, I think, whereby the current annotation was just too short. And you can see that also that some of the orthologs have the longer version, the Ostra, for instance. So this gene here looks perfectly normal. Introns are correct. You can see here, it's UTR is probably too long, but you wouldn't think that something is incorrect. But if you have a closer look, you notice that this should be actually on the starting on another frame. And Apollo shows that quite nicely with different colors. And in fact, if you got it on the correct frame and you analyze it, you get orthologs in Plasmodium, and the Plasmodium gene has been analyzed and experimentally verified, and I've given it a name. And so the Toxoplasma gene also now is called protein MPOEDD. We've also got genes that are not present currently. And here's an example. As you can see here, this gene nestled here in this UTR region, which is too long. And you can see here it's on this strand. If you run interproscan, it comes up as cytochrome C oxidase. So a small one exon gene has been added. So we've got over 250 new genes that were added. And so another example here is a new gene. The old gene was predicted on the incorrect strand, and it's supported by evidence. And in fact, it has been analyzed and it has been experimentally verified in a publication from 2009. So clearly, this one is the correct one and got now a proper product. We've got 140 new merged genes, and you can see here that we've got 3 merges, nicely supported by evidence. We've got split genes, I think only 20, but so this is 1 gene, and you can see here quite nicely, this is supported, the split, and also shows here that there is a possible long non-coding RNA in the middle of those 2 genes. And this split has also been mentioned and analyzed here in a publication where they've looked at protein phosphatases. And I mentioned and looked at that this gene should be split. So this is the protein phosphatase now, and this is a hypothetical protein. David already showed you an isoform. We've got a little bit over 20 isoforms now, and here is an exon skip, quite nicely supported. Plus, it has also been mentioned in one of these, in a publication from Stuart 12 about isoforms. So he had this gene also as example. And finally, obsolete genes. So we've got here genes that are not supported anymore. These 4 little genes are on the wrong strand. This should be is the correct annotation on the correct strand. And yeah, so these are on the list of 260 something genes that should be deleted, and they have no evidence.
I mean, it's worth pointing out the shades of gray here. So, for example, this protein most to the left is one of the introns of that is in fact supported by evidence, but only with 3 reads. So there's very low abundance transcription there. Certainly nothing we would want to annotate as a gene in the basis of existing evidence. In contrast to the read that is highlighted on the opposite strand, or the transcript that's highlighted in the opposite strand, which is supported by hundreds of reads.
Quick question: how much of your transcriptomes are strand specific?
Many. I mean, so I don't know whether it's the majority, but probably for Toxoplasma, I think it's almost certainly the majority. Certainly the majority of the best data is strand specific. But keep in mind that that while overall transcript data is difficult to interpret if it is not strand specific, intron spanning reads are very informative even from non-strand specific data because of the non-palindromic nature of the intron conserved intron junctions.
Okay, so these are all the structural changes examples I wanted to show you, but we not only, and David already mentioned that, we've got hundreds of product changes, many gene symbols were added and Go terms, and this is based on the many user comments that have been added over the years. So they're incredibly useful. So please keep them coming in because they can be used to update functional and structural annotation. And 2 examples of a functional annotation is shown here. So you can find them on the gene record pages in the annotation, curation, and identifier section, in the community annotations from Apollo track. And what you can see here is the new product, and also the support, the PubMed ID that supports it, gene symbols and also Go terms can be seen. And David already highlighted that on the gene record page, you can find all of those changes in the community annotations track from Apollo in the JBrowse window. And if you click on them, there is a pop-up where you can then see the product, if this gene has a product annotated in Apollo. And in this case, this is a new gene. And you can see here, this is a selenoprotein, and it also has been mentioned in a publication. And that's all I want to say in terms of Apollo and examples on current changes. Thank you.
One thing to, thanks, Uli, that was very informative. One thing that I'd like to point out is that I think most of you here are aware of our user comment system, where you can go to any gene page, and as long as you're logged in, you can quickly add a user comment on a gene. You know, something like, you know, this gene is not hypothetical, it's a phosphofructokinase, right? That could be a user comment. The user comments actually serve 2 purposes. One is to collect things like annotation and information about the gene function, but also to collect your expert knowledge, which could be anything from information that will modify a potential gene name to information that you have that may not go anywhere. Like, I tried to knock out this gene 10 times and it never worked, so maybe it's essential. That's very useful information. Or you can imagine many different ways of doing that. What Uli showed you in Apollo is a method to add your functional annotation as well through Apollo, and that gets directly integrated into the Apollo system for updating the annotation. And so that the user comments are manually reviewed, not always, but because you need a person to actually manually review them, and it takes effort. Whereas the Apollo annotation is a bit more automated in terms of its integration. And Uli, correct me if I'm wrong if I misspoke about this, but those get, once you click on finish, those get integrated as a community annotation as well. And then and so one big goal of this webinar, as we talked about, is to tell you about sort of the plans for what to do next. So right now we have, as Uli showed you and David showed you, we have all of these gene models and annotation changes that are available in the community annotation track. Those were provided, a lot of them were provided by Uli's expert work, but also some of you in the community have added those, which is great. Now the question is what are the next steps? What do we do with those? How do we make those part of the official annotation? And of course, we mentioned all the caveats. So before we go on, maybe I want to see if there are any specific questions from attendees. So there's question, do we have any gene prediction tool for cytosine or Babesia species on your Galaxy server? Certainly we have the tools installed in the Galaxy server for just like any other Galaxy server, we have multiple bioinformatic tools. We would have to go in individually and see which tool you're interested in, see if it's installed, and then give it a try.
Yeah, but that does raise an important point. The ViewPathDB Bioinformatics Resource Center supports many hundreds of species. We, because of the available data and the historical engagement of the Toxoplasma community, we have focused this workshop this webinar on Toxoplasma, and we would like to devote the coming weeks to small number of months to capturing expert knowledge from the community in a way that can be communally to update the reference genome with the best available information on gene models. But the same kinds of modifications can be applied to any reference genome, including for Babesia. And we are also happy to work with individual labs and with communities, for example, in connection with the upcoming Babesia meeting that ViewPathDB will be present at and support, to ensure that this information is, that similar updates are available for Babesia or Aspergillus or Anopheles or whatever other organisms may be of interest to the community.
So let's pause for a few seconds and allow people to ask questions if there are any questions. Okay, so in terms of timeline for updating the official annotation, what we were talking about internally is providing the community. Obviously, these community annotations are there. They're already in ToxoDB. They've been in ToxoDB for a while. So we encourage everybody to go to your favorite gene and look at it, see if there's a community annotation provided. And if there is, see how it changes your gene. And if it changes it to the better and it's accurate, then that's great. If it doesn't, then we highly encourage you. In fact, it's almost a must that you go in and edit the gene yourself in Apollo, or if you need help, email us or contact us and just say, hey, you know what, I'd like to do this. I'm not able to figure out Apollo. Can we have a quick Zoom and show me how to do it? And we would be more than happy to do it. So the timeline that we're looking at is to for the current community annotation that's available to, and anything that's added from now until the deadline, which we're proposing to make it June 1st. And that is basically, and we'll basically lock that community annotation on June 1st, and then work on integrating that into the official annotation of the current ME49 genome. That's one option. That's one timeline we are considering. Obviously, people can continue adding community annotation even after we lock it down. It's not meant to say that, okay, no more community annotation. It's just that we have to stop things at a certain point to be able to start working on updating the official annotation. So, of course, built into that is the issues with the assembly. And that's where I think it would be great to hear from people and get some opinions. We, of course, can come up with our own conclusion and decide what to do. And if you're happy with that, and you're happy to give us the approval, that's great. But of course, it'll be great to hear from you, as many of you are experts in this and have probably some very valid opinions about this.
So it's also worth pointing out, Omar, a note in the chat from Mark-Jan Goebbels, a anonymous comment from him, is an anonymous reviewer for many publications. Mark-Jan is pointing out the importance of communicating to the community that peer reviewers on publications should be sure to point out that publishing a paper that describes, for example, the modification of the correct identification of a translational start site or alternative transcripts or inaccuracy of gene models really doesn't constitute finishing your publication until that information is appropriately represented in ToxoDB. So I think Mark-Jan is suggesting that those who are involved in writing such papers be sure to make sure that their changes are reflected in the reference database, and that those who are responsible for reviewing would request such changes to be made in association with reviewing a paper. Mark-Jan, feel free to unmute yourself if I've misrepresented your views there in any way.
No, David, that's the accurate description of what I was trying to say. Yep. And
I think we've answered Praveen's question about cytoxoan or babesia. So Omar has highlighted the proposed timeline. So I'd like to encourage everyone on this call, everyone in the labs that you represent, and all of your friends and colleagues, including those who we trust will be viewing this recorded webinar asynchronously. We apologize to those who are in California for whom it's pretty early in the morning, but obviously making it later in the morning for people in California would make it unacceptably late at night for those dialing in from South or East Asia. So our proposal is to try to to complete this round of updates by the end of May, and make sure that information is then reflected in the reference genome. That does not mean that June 1 is the last moment that you have to make sure that any information is accommodated. We will continue to support the Apollo platform, and additional annotations can be made at any time. But that does not mean that, and it will be reflected in the Apollo annotation track. But we would like to make sure that we have a comment period then to allow people to note any changes on their favorite genes and dispute anything that they disagree with, and make sure that information then is reflected in the reference, the reference gene annotation. That does so I hope that we've touched on why this is needed. There's much opportunity to update the already outstanding annotation for the Toxoplasma genome to make it even better, and the opportunities for training and support. That leaves aside the issue on how or if one updates the reference genome, and I'll turn back to that in just a moment. But I see that Andy has his hand up. Anything you wanted to add?
Yeah, just a quick comment to say, maybe you're going to come on to this, but also to sort of say about after that 1st of June sort of cut off and update the reference genome, we also would want to consult on the strategy for what happens to the other strain genomes. Were you going to come on to say a little bit about that?
I was, because that's an important issue about what the reference genome is. So our preference is, and do feel free to raise your hand or contribute to the chat if there are opinions on this from the community, as I'm sure there must be from the scores of people who are currently on the call. Once we have updated the reference genome, that does raise the question of what our reference genome consists of. And as Omar has indicated, we are certainly aware of dramatically improved reference genomes for ME49 strain from at least a couple of labs, and from other strains as well as for the Neospora genome. With long read data, it spectacularly enhances the ability to close gaps, including in telomeric regions, and to properly reassemble areas that are currently shown as having copy number variants that are in fact compression zones of typically tandemly repeated genes, or cases of multiple paralogs distributed throughout the genome. There are others on this call who are more familiar with this than I. We do need to determine which genome to select as the reference, and we will of course consult with the community, but I think everyone would be better served by an improved genome. If you have views on that, please make them known here or offline, preferably through clicking on the Contact Us link so it enters into our tracking system. And once that decision is made, we also need to think, as Andy has mentioned, how or if this information should be reflected in other strains and other independently annotated genomes. Omar, I see your hand up.
Yeah, just quickly to, in case people are not aware, we did load, I think over a year ago, one of these nicely assembled genomes, and it's an RH strain, RH88, I believe, from Hernan Lorenzi, and that's a genome that is assembled with the right chromosome number, and it has its own annotation. Of course, it's a separate annotation, but you can view it in synteny or collinearity with ME49 or VEG or whatever genome you're interested in, and you can visualize and see where the changes are. So that's a nice way to do that. In terms of the other genomes that are sequenced from Maria Francia and John Boyle, those are currently only available as assemblies in GenBank. The annotation is not available, which actually may end up being a good thing, because it means that we can maybe now focus on making sure that we integrate a unified annotation for all of these, instead of populating the database with 20 other versions of annotation of the same strain.
So thank you. So thank you, Omar. And Andy raises an important point. What this brings us on to. So currently we have semi-independent annotation for, I think, 16 Toxoplasma strains. And I suspect that most of us on this call have been excited to explore that, to try to identify the potential molecular basis of the very important biological differences between some of these strains. And not surprisingly, what we have discovered is that the vast majority, well over 90% of the variation in the annotation between strains is artifactual, is a function of inappropriate transfer or annotation of those individual genes. No one disputes that there is difference between strains, for example, in the number of tandemly repeated rope kinase genes in rope 4-7 locus or rope 5 locus. But if you identify a gene that has a different UTR in the VEG strain compared to the ME49 strain that you think might be associated with alternative transcription initiation sites and transcription factor binding sites, it's almost certainly an artifact of annotation rather than a genuine difference between the strains. If, for example, you were to go to look at the human genome at the UCSC browser, you will go and look at the human genome, not the Claire Harding genome or the Michael Reese genome, since their names are showing up in front of me on the on my Zoom panel view. You're going to look at the human genome, even though we're certainly aware that there are differences between the Michael Reese genome or the Claire Harding genome or the Craig Venter genome. So I think our recommendation is that we represent all variation as by comparison with a single reference assembly, probably, but not necessarily from ME49, but that among many other questions require advice from the community. Andy also asked about how this information is transferred over to other genomes, and if the decision is that it's important to maintain more than one reference, more than one annotated genomes, hopefully not 16. That's something that we need to think about how to lift over this information and update the changes, for example, that Uli just made today live onto the VEG genome and the RH88 genome and so on. But my personal strong recommendation is that we select one Toxoplasma genome as we do one human genome, represent other differences as differences on that reference, and also importantly preserve the historical information that is currently available on those other genomes by lifting over user comments and providing an annotation track that maps the current annotation, say, provided by Hernan Lorenzi and JCVI on the RH88 assembly as another annotation track that's available mapped to ME49 or whatever the new reference is. So
we
this
are
is a topic for another. This is a topic for another day, and we will have some further discussion. But people may have comments or questions that they want to ask, and I know, Omar, that we are down to our last couple of minutes. So let's make sure that we have time for any questions. And I'll point out that there are also many other functionality associated with ToxoDB and ViewPathDB that you may be interested in clicking on the Contact Us link to follow up on offline.
So, of course, if you have any comments right now, feel free to put them in the chat or unmute yourself. But of course, we'll welcome emails from you after this webinar, after you think about the questions we asked. We will obviously follow up with a summary email and a link to this recorded webinar to the Toxo community with information about the timeline as well, so we can solicit more feedback. Right now, if you don't feel like talking or saying anything, feel free to give a thumbs up or thumbs down using the reactions. I'm not sure you can do a thumbs down, but you can definitely do a thumbs up. And in case you are, you're happy with sort of the plan in place. And again, obviously, the more we hear from you, the more you tell us about not only the annotation, but also features that you think the database should be providing or data sets that are missing that should be there. Definitely let us know. We're always happy to hear those so we can improve this community resource.
John, go ahead.
Yeah, so a question I have is, once you do this update, you then send the information to AlphaFold, to
Yep.
NR database, all those kinds of things, because it's extremely important now that everybody's using FoldSeek to search the world to have wrong things from
True enough. So that's one of the many reasons to ensure that the information which is currently represented in user comments or in the Apollo annotation track enters into the official annotation, so that it does wind up being being sent to AlphaFold. So currently what happens, for example, we do, as I'm sure you know, provide AlphaFold structures for all genes in ViewPathDB. Currently, any information that is represented in a user Apollo track will not have an will not have a gene page and a distinct gene page, and will therefore not have an AlphaFold structure. Once that is loaded into ToxoDB, it will. And once that has been synchronized with a referenced annotation in GenBank and Ensemble and INSDC databases, which is a longer timeline that is not entirely within our control, it will also be more obviously accessible to the broader world.
Okay, if there aren't any other questions or comments, thanks everybody for joining. And again, please think about what we talked about today. If you have anything you want to contribute, drop us an email, and we're happy to obviously have a discussion about any of your ideas. Thanks everybody. Thanks David and Uli, and we'll see you all next time. All right, bye-bye.